Hello, and welcome to this Nature.com custom webcast titled Applications of Analytical Light Scattering in a Biophysics Core Facility. My name is Gyration Karpen, and I will be your moderator. Today's webcast is sponsored by Wyatt Technology Corporation. Wyatt Technology is the recognised leader in instrumentation for determining the absolute molar mass, size, charge, and interactions of macromolecules and nanoparticles in solution. We'll begin today's webcast with a presentation and end with a Q&A session where we will address the questions that have been submitted by you. To ask a question, just type it in where it says type your question here and then press submit at any point during the webcast and we will answer them today. It is now my pleasure to introduce today's speaker, Dr. Eva Folter-Stognew, who is the Director of the Keck Biophysical Resource at Yale University School of Medicine. Over to Eva.
Jason, thank you very much for your introduction, and welcome everybody to the webinar Applications of Analytical Light Scattering in a Biophysics Core Facility. All the data I will be presenting today were collected at Biophysics Resource of Keck Laboratory at Yale School of Medicine. The mission of the resource is to provide biotechnology support that allows quantitative characterization of interactions between biomolecules using in-solution biophysical method. We assist investigators in measurements of binding affinity, stoichiometry, kinetics, and thermodynamics of complex formation. To assist in these measurements, we house multiple technology, among those light scattering instrumentation, microcalorimetry, CD spectrometry, subflow spectrometry, and surface plasmon resonance biosensing. Out of all this technology, today's webinar will only talk about the results collected utilizing light scattering, either static or dynamic, which are in line with the size exclusion chromatography. I will now go into details of the physical phenomenon that govern the application of static light scattering to measure molar mass, or how data collected by dynamic light scattering detector can be used to measure hydrodynamic radius. I will discuss strictly the case studies and how these technology have been used in this core to answer biologically relevant questions. The SECMOL service provided by the core uses the setup that combines light scattering measurement with either size exclusion chromatography or flow-field flow fractionation. The fractionation on the sizing column or FFF channel is used purely as a separation step to resolve into individual fractions various components of complex sample. There are multiple detectors in line with the fractionation column that allow collection of absorbance signal, static and dynamic light scattering signal, and finally measure changes in refraction by refractometer. Refractometer is particularly critical because it serves as a universal mass detector that allows detection of elution of sugars, lipids, or other moieties that do not contain any spectroscopic signal. This is the photo of the actual system that we house, which contains the Agilol 1200 HPLC system with diode array detector and fluorescence detector, and Wyatt's Donhelius 2 static and dynamic light scattering detector, and Optilar Rex refractometer. The results collected in the SECMOL's analysis are reported in so-called molar mass distribution plots. These plots contain a typical fractionation trace, which is either UV absorbance at 280 or 220 nanometers, or a trace recorded by refractometers. These are actually individual dots which represent molar mass measurements across the elution profile performed every 2 microliter during the elution of the sample. For this particular sample, which is a BSA sample, the major mass corresponds to the molecular weight of 66 kilodalton in agreement with the monomeric form of the protein. The small peak that elutes ahead of the main mass contains molecules with molar mass of 135 kilodalton, which is corresponding to the dimeric form of the BSA. And you will be seeing a lot of those molar mass distribution plots in my presentation. So one can ask the question whether this is all that we can learn from the SECMOL's analysis. The aim of today's presentation is to provide a glimpse of the wealth of information one can derive from secmoles analysis, beyond just measurement of molar mass. I hope that the data presented will illustrate how the secmole service provided by the biotechnology core can advance diverse research projects. The core has performed thousands of secmoles analyses, and it was a difficult task to narrow down the presentation into just a few case studies. But I hope the examples I selected will illustrate how secmoles analysis can answer biologically relevant questions, like determination of stoichiometry for protein nucleic acid complexes or membrane proteins that are complex with lipids and detergents, how the combination of static and dynamic light scattering can be used to analyze shape and morphology of protein assemblies, and finally, how we can determine dimerization constant from secmoles measurements. So let's start with the studies of stoichiometry of protein nucleic acid complexes. FIR protein is a repressor involved in regulation of transcription of c-myc oncogene. The research was approached with the question, what is the oligomeric state of this protein in solution? Even though FIR protein was seen as a dimer in the crystal structure, in solution we could not detect anything but the oligomeric form that corresponds to the monomeric FIR with the molecular weight around 24 kilodalton in agreement with the monomer of FIR. We analyzed the protein in the concentration ranging from 0.1 to almost 5 mg per ml, and even at 5 mg per ml, the major form was the monomer. This result was so surprising that the oligomeric state of FIR was also analyzed by analytical ultracentrifugation. And as illustrated in this graph, in pink are the results from AUC analysis, while in blue are the results from secmoles analysis. As we can see, both technology observed mainly monomeric form of FIR, with the difference being that AUC analyzed the protein only up to 2.5 mg per ml, while by secmoles analysis we reached almost 10 milligrams per milliliter. However, even at this very high protein concentration, the major component is a FIR monomer. The next question in the project was to measure oligomeric state of the protein DNA complex. The protein was crystallized complex with the fuse element, which is an 8 kilodalton fragment of single-stranded DNA. As illustrated in this molar mass distribution plot, the elution of the protein complex varies with changes in protein concentration. In particular, once the protein DNA complex concentration increases, the protein elutes in the earlier elution times. Similarly, with increasing protein concentration, we see increase in the molecular weight at the apex of the eluting peak, with the FIR DNA complex reaching 58 kilodalton at the highest protein concentration tested. Please note that even though the measured molecular weight is only 58 kilodalton, this protein DNA complex elutes ahead of the 66 kilodalton molecular weight standard, showing abnormal elution position for this particular protein DNA complex. When observed molar mass is plotted as a function of FIR protein concentration, we can see that FIR protein alone remains monomeric in solution, while binding to the DNA induces protein dimerization. The observed molecular weight at the highest protein concentration we tested, which is 58 kilodalton, falls somewhere in between the two stoichiometries that we can expect for the FIR DNA complex, namely the one piece of single-stranded DNA bound to the dimer of FIR, or two pieces of single-stranded DNA bounded to dimer of first. The observed molecular weight is somewhere in between, so to narrow down the stoichiometry to either 2-to-1 or 2-to-2 complex, we use another piece of information that we can derive from secmoles analysis. We can utilize the fact that the DNA molecule is a very highly UV-absorbing molecule in comparison to polypeptide. Thus, the measured UV to RR ratio for DNA is 2.34, while the UV to RR ratio measured for the FIR polypeptide is only 0.14. Because the observed UV to RR ratio for the DNA fragment used is 17 times higher than that of FIR protein alone, the FIR DNA complex with 2:2 stoichiometry is expected to have much higher UV to RI ratio than 2:1 complex with only single DNA piece bound to the dimer of FIR. At the highest concentration tested, we measured the molecular weight that falls somewhere in between these two stoichiometries. However, the UV to RR ratio measured matched perfectly the ratio that we would expect for the complex that is built from dimer of FIR associated with the single piece of DNA. Thus, combining the two pieces of information derived from SECMOL's analysis, namely the molecular weight we measured and the UV to RR ratio we observed, we can with confidence say that the complex that FIR DNA forms is a dimer of FIR associated with single piece of DNA. Interestingly, when we look at the complex that we observed at 4 micromolar concentration, this complex has molecular weight of a single monomer of FIR associated with a single piece of DNA. And interestingly, the UV to RR ratio we observed matched perfectly such 1:1 stoichiometry. So this suggests that the assembly process is binding monomeric FIR to single-stranded DNA, and then the second molecule of FIR is recruited for the FIR DNA complex, and the FIR protein dimerized while binding to DNA. Just in summary, the SECMOL's analysis for FIR alone and FIR in the presence of DNA indicates that the FIR protein has propensity to dimerize, but this propensity is observed in solution only when the FIR is associated with the piece of DNA. So even though we see dimeric form of FIR in the crystal structure, in solution the protein will only dimerize when it starts binding to the DNA. The second case studies I would like to discuss refer to utilization of SECMOLs for analysis of integral membrane proteins. Such proteins are purified in the presence of detergents and are solubilized as protein detergent complexes with unknown amounts of detergent associated with the polypeptide. Determination of the oligomeric state of such protein detergent complexes is challenging because these proteins often behave anomalously during size exclusion chromatography. During analysis of modified proteins, that is, proteins that contain polypeptides and other non-polypeptide moieties like detergents for membrane proteins, sugars for glycoproteins, or polyethylene glycol for pegylated proteins, the SECMOL's data are processed using the so-called 3-detector approach. This method was initially described in detail by Tanaki Group in Japan for analysis of membrane proteins, and later on applied by John Filer Group at Amgen to analyze glycoproteins and their complexes. The 3-detector analysis links the responses recorded by 3 detectors, light scattering detector, absorbance detector, and refractometer, to molecular weight of the polypeptide portion of the protein complex. The calibration of the system is conveniently obtained from responses collected in the 3 detectors during analysis of protein standards of known molar masses. The elegance of this approach lays in the ability to use unmodified protein to construct the calibration curve that later can be used to determine oligomeric state of modified protein. Thus, even though simple proteins are used to calibrate the 3-detector system, such calibration allows determination of oligomeric state of polypeptides that are glycosylated, pegylated, or associated with lipids and detergents. Hence, the responses are recorded in the 3 detector. The ratio, 3 detector ratio, is computed, and this value is later on used to determine the molecular weight of the polypeptide portion directly from the calibration curve. Currently, this approach can be applied directly during processing SecMol's data, since the 3 detector approach has been incorporated into Astra software. So the software records the responses in the absorbance, refractometer, and light scattering signal, and solves the sets of 3 equations to compute the molecular weight of the polypeptide portion and the molecular weight of modifier. We have to know extinction coefficient for both polypeptide and modifier, and the D and DC value, so-called refractive index increments, for polypeptide and modifier. And once we input this known, then we can directly compute the molecular weight of the entire complex, the polypeptide portion for it, and the molecular weight of the modifier. The examples that I will be discussing, however, were analyzed way before the conjugate analysis was available, so they were processed using the 3 detector approach that I just described. These were the first 2 proteins that the resource analyzed in 2003 as proof of principle before we applied this method to determine trimeric subunit stoichiometry for glutamate transporters. These 2 proteins were supplied by Eric Guek's lab. The crystal structure of these proteins were already solved, so we expected that the porine will be trimer with the molecular weight of 141 kilodalton, while the hemolysin would be a hectamer with the oligomeric molar mass of 231 kilodalton. Both proteins were purified in the presence of the same detergent, the decyl maltoside, that was present at the concentration that is few times higher than the critical micelle concentration. This slide shows the 3 signals collected during SecMol's analysis of porine LAM-B. In the elution profile, we can clearly see that in the UV trace, shown in blue, there is a single peak eluting at around 15 mils, which also generates responses in light scattering detector, shown in red, and refractometer in green. Interestingly, we can detect the empty micelles that are clearly visible in light scattering trace in red and refractometer trace in green, while generating known signal at the absorbance at 280 nanometer. When we apply the 3 detector analysis, we can see uniform molar mass across the eluting peak that corresponds to 141 kilodalton, which is in perfect agreement with the molar mass expected for a trimeric form of this protein. When we apply the same 3 detector analysis for SecMol's data collected for hemolysin, we computed the molar mass of the polypeptide as 225 kilodalton, in close proximity to 231 kilodalton expected for a heptameric form of the protein. We observed an interesting behavior during this testing, namely, when the UV traces of these 2 proteins were overlaid, we can clearly see that the trimeric porine with the molar mass of 141 kilodalton elutes ahead of the heptameric hemolysin with the molar mass of 225 kilodalton. So why is the smaller trimer eluting ahead of the larger heptamer? To answer this question, we can utilize another information that we can obtain from 3 detector analysis. We can use the signals recorded by refractometer and absorbance detector to compute the amount of the detergent that is associated with the polypeptide. Because lipids and detergent usually do not absorb at the UV signal represents the concentration of the polypeptide, while the signal created by refractometer measures the concentration of the entire complex, namely polypeptide, lipids, and detergent. So between the 2 signals collected by UV and refractometer, we know the concentration of the protein and the concentration of the modifier. So we can do the math to compute how much modifier is associated with the polypeptide. When we perform this analysis, we learn that the porine binds over 1 gram of lipid per 1 gram of polypeptide, and the entire complex of polypeptide and detergent has molecular weight of 285 kilodalton, while the hemolysin binds only a little bit over a quarter of the gram of lipids per 1 gram of polypeptide, so the entire complex has molecular weight of 271 kilodalton. So this information explains why the trimeric porine elutes ahead of the heptameric lysine, because the overall complex for the porine is larger than the complex formed by the hemolysin. So, in summary, for detergent solubilized membrane proteins, elution from size exclusion chromatography is governed by the overall size of the protein detergent complex and may not correlate with the oligomeric state of the polypeptide itself. Thus, the trimeric porine with molar mass of the polypeptide of 141 kilodalton elutes ahead of a heptameric hemolysin with molar mass of 225 kilodalton. These examples illustrate clearly the triple detection by light scattering, absorbance, and refractometer is required to properly characterize such protein detergent complexes, as SECMOS analysis allows determination of oligomeric state of the polypeptide regardless of its elution position and the amount of detergent associated with it. And SECMOS will provide means to determine the amount of the detergent that is associated with the polypeptide and thus allow computation of the molar mass of the overall polypeptide detergent complex. I will now tell you a story how we used a combination of static and dynamic light scattering to study morphology of protein assemblies. The protein under investigation is PopZ protein, which is bacterial scaffolding protein involved in cell division. We were approached to identify oligomeric state of the wild-type form of the protein and 2 mutants that were of interest because of their biological effects. PopZ protein is a 178 amino acid protein with a monomeric molar mass of around 21 kilodalton. In native gel, however, the protein runs with apparent molecular weight of 600 kilodalton. The 2 mutants of interest were C-terminal deletion mutant, marked as SN. This mutant was selected because it preferentially assembles into a slower migrating form similar to the wild-type protein. The second mutant, marked as PA, is a single point mutation proline to aline substitution at position 146. In contrast to SN mutant, the PA protein assembles exclusively into fast migrating form with apparent molecular weight of 240 kilodalton. The initial SECMOL test revealed that all 3 versions of the protein elute very early from the sizing column, eluting ahead of the 475 kilodalton apoferritin. Despite such early elution, the protein molar masses were rather small, ranging from 60 to 200 kilodalton. These molar masses measured from SECMOL's analysis were significantly smaller than the apparent masses estimated from native jet electrophoresis of 600 kilodalton for the slow migrating forms of wild-type NSN, or even the 240 kilodalton estimated for the fast-migrating PA mutant. The very early elution observed for the PopZ protein suggests elongated shape. To confirm this idea, we use dynamic light scattering to measure the hydrodynamic radius of PopZ protein. The DLS measurement revealed that these are hydrodynamically very large proteins. Despite their modern molar masses, the hydrodynamic radii were measured as 6.8 for the trimeric PA mutant to 11.5 for wild-type protein that form structure larger than hexamers. It is rather remarkable that the 71 kilodalton trimer has the size that is comparable to a 475 kilodalton apoferritin. The knowledge of molar mass and the hydrodynamic radius for a protein allows determination of frictional ratio, marked as F over F0. Frictional ratio designates the ratio of the frictional coefficient of a macromolecule to the frictional coefficient of a compact sphere with the same mass and density. The greater the frictional ratio, the more asymmetric the protein is likely to be. We can clearly see that PopZ protein forms very elongated structures with frictional ratios above 2. Typical globular proteins have frictional ratios of 1.3, and this is exactly the number that we've measured for apoferritin. The frictional ratio that we observed for PopZ proteins were so unusual that we performed CD analysis to confirm that these proteins are folded. The CD spectra revealed that the central portion of PopZ protein contains left-handed polyproline fold, so-called PP2 fold, which is characteristic for natively unfolded proteins, and the presence of this unfolded portion of the protein explains the large hydrodynamic size and the large frictional ratio that we observed for this protein. Additional secmal analysis revealed that at the concentration tested, the mutant proteins appear to retain oligomeric state, with mainly trimeric form observed for the PA mutant and hexameric form observed for the SN mutant. It was also clear that the wild-type proteins form structures that are larger than hexamers. We have, however, noticed that while the SN mutant retains elution position that is not affected by changes in protein concentration, the elution of the PA mutant shifts to earlier elution volumes with increasing protein concentration, suggesting that there might be concentration-dependent changes in the shape of the protein. This observation was in line with the behavior observed in transmission electron microscopy, which shown that the purified PopZ protein forms elongated filament for the wild-type protein and the PA protein, while the SN mutant was found to be defective in filament formation. The CD analysis of PopZ protein confirmed that the PA mutant interrupts the PP2 fold present in the central portion of the protein, while the SN mutant is missing critical region of the helical portion at the C-terminus. The basic building block is an elongated trimer easily formed by wild-type and mutant proteins. Lateral interactions facilitated by the PP2 regions of the protein are necessary to form hexamers. Thus, the PA mutant in which the PP2 fold is interrupted has less propensity to form hexamers. Mutation in the C-terminal domain also blocked discrete steps in the assembly of higher-order structure. So at the concentration where the wild-type protein forms higher-order oligomers, the SN deletion mutant remains hexameric, confirming that the final 6 amino acids at the C-terminus of PopZ are necessary for connecting the hexamers into filaments through head-to-tail assembly. While the PA mutant is deficient in hexamer assembly, it is still capable to form filaments. Thus, in summary, the biophysical analysis of purified wild-type and assembly-defective mutant proteins indicates that PopZ self-associates into an elongated trimer, which self-associates into hexamers, and finally the hexamers forms the filaments through head-to-tail assembly process. The final portion of the presentation will illustrate how SecMol's data can be used to measure dimerization constant. For some proteins subjected to SecMol's analysis, we observed that the measured molecular weight increases with increasing concentrations, as illustrated here for nucleobinding 1, a protein with monomeric molecular weight of 51 kilodalton. In the few analyses selected for this graph, we can observe that the molecular weight varies from 50 kilodalton to roughly 100 kilodalton, suggesting that the protein is monomeric at low concentrations, and as the concentration increases, the protein starts forming higher order oligomers. When we subjected this protein to SecMol's analysis at a wide range of protein concentration, ranging from 1 microgram per milliliter to 1 milligram per milliliter, so spanning 3 orders of magnitude in protein concentration, we observed that the molecular weight at the apex of the eluting peak changes from 50 kilodalton, which corresponds to a monomeric form of the protein, and plateaus around 100 kilodalton, which corresponds to a dimer. We also observed that the measured molecular weight was independent on starting protein concentration, but rather was related to the concentration of the protein at the eluting peak. At the apex of the eluting peak, we can select 5 to 10 measurements in which the protein concentration remains constant, so we believed that these slices contained a mixture of monomeric and dimeric form of the protein that are in equilibrium governed by dimerization constant. To confirm this assumption, we analyzed the protein at 2 different flow rates to test whether, regardless of the time that the protein spends during fractionation step, the measured molecular weight will be only dependent on the final eluting concentration. And this is exactly what we observed, regardless whether we measure the protein molar mass at half a mil per minute or 1 mil per minute. The molecular weight is only dependent on the concentration, not on the time that it takes to analyze the protein. We know that static light scattering measures weight average molecular weight, which for dimerizing protein rests on the distribution of the protein mass between fraction of the monomer and dimer. The partition of the protein between monomer and dimer is governed by dimerization equation and dimerization constant. Thus, at a given protein concentration, the fraction of the monomer is solely dependent on the dimerization constant. Thus, the measure's weight average molecular weight is only a function of protein concentration and dimerization constant. Thus, fitting the observed weight average molecular weight as a function of protein concentration allows determination of the dimerization constant. We use this approach to study the effects of N-terminal deletion on the dimerization constant of Sec-A protein, which is nanomotor promote that is involved in protein translation in eubacteria. Based on the crystal structure, the N-terminal portion of Sec-A is involved in dimer interface. Thus, the 2 deletion mutant that are lacking 8 or 11 N-terminal amino acids were subjected to dimerization constant analysis along with the wild-type protein. Because there were inconsistent reports in the literature about the stability of Sec-A dimer that seemed to be related to buffer condition used, we tested the dimerization at low and high salt concentrations. It was evident from the initial tests that the increase in the ionic strength causes the destabilization of the dimers for both the mutant's protein and the wild-type protein. While we still have some dimer of the wild-type protein left, the D11 mutant appears to be completely monomeric at high salt concentrations. When we look at the full concentration-dependent of the molecular weight for the D11 mutant, we saw no changes in the molecular weight at the concentration range we tested. When we performed the same analysis at the low ionic strength buffer, we could see strong dependence on the molecular weight when the protein concentration is increased. These pronounced changes in the molecular weight as a function of concentration can be used in the approach that I just described to derive the dimerization constant for sec-A protein. We use this approach for analysis both in low ionic strength and high ionic strength for all three proteins tested, and we can see that moving from low ionic strength to high ionic strength destabilizes all proteins, with the D11 mutant remaining almost monomeric during the entire concentration range tested, while at low ionic strength the protein still dimerizes. When we convert dimerization constants into Gibbs free energy changes, we can perform the thermodynamic linkage analysis of dimer stability. Thus, we know that dimerization of wild-type protein is driven by over 12 kilocalorie per mole change in free energy. We also know that both deletion mutants form less stable dimers, with free energy change of 7.4 and 9.7 for D11 and DS mutant respectively. Interestingly, we also measured that increasing the salt concentration from 100 millimolar to 300 millimolar salt is more destabilizing than removing 8 amino acids from the N-terminus. Removing 11 amino acids is more destabilizing than changing the buffer condition. In summary, we confirmed that as expected from the crystal structure, the sec-A dimer can be destabilized by removing N-terminal amino acids that form the dimerization interface. The sec-mol analysis illustrated, however, that comparable destabilization of sec-A dimerization can be accomplished by increasing ionic strength by elevating the salt concentration to 300 millimolar. The final project I would like to discuss studied dimerization of extracellular ligand binding domain of the metabotropic glutamate receptor. From the initial analysis, we could see that the mutation that was introduced was destabilizing to the dimeric protein. The major challenge in this project was rather poor yields in protein preparation. The protein was expressed in the hex cells, and a single preparation yielded only 25 macrogram of proteins. We analyzed this protein from 3 different preparations, and the results are shown with different symbols representing data collected from various protein purifications. The results shown on this graph represent analysis at 3 different flow rates to confirm that these measurements represent the protein in monomer-dimer equilibrium. The results revealed that even the destabilized dimer is rather stable with dimerization constant of 28 nanomolar. Because the dimer was so stable, we needed to measure the molecular weight at rather low concentrations. The lowest point on this isotherm represents measurements at concentration that is below 1 microgram per milliliter. As you can see in the signal for light scattering detector shown in red and the refractometer shown in blue, even at this very low concentration, there is a remarkable robust signal-to-noise for both of these measurements. We also use sec-mols to illustrate that binding of glutamate stabilizes the dimer and shifts the dimerization constant into single digits nanomolar value. The case studies I discussed today illustrated that a single analytical sec-mol system allows measurements of molecular weight and the concentration extending from 1 microgram per milliliter in case of dimerization of nucleobinding or glutamate receptor to concentration ranging to 10 milligrams per milliliter in case of studying dimerization of FIR protein. This represents concentration range that spans 4 orders of magnitude, that is accessible for determination of molecular weight on a single secmoles analytical system. I hope that the study cases I discussed today illustrated that size exclusion chromatography coupled with light scattering allow fast and accurate determination of molar masses in solution, that this approach is a critical tool for characterization of non-globular macromolecules, protein complexes, especially complexes that elude abnormally from sizing columns or show unusual mobility during native gel analysis. This approach is suitable for a wide range of protein concentration ranging from 1 microgram per milliliter to over 10 milligrams per milliliter. I also showed that the secmoles DLS data are very information-rich and can be utilized to determine the stoichiometry of protein complexes, provide information about the shape of the protein assemblies, as well as be used to measure the dimerization constant. I would like to thank NIH for funding the secmoles instrumentation, Biotechnology for ongoing support with the detectors, users of the Biophysics resource who entrusted their samples for secmoles analysis. I would like to thank you all for your attention and invite you to submit samples to see for yourself what you can learn about your project from secmoles service. Thank you.
Thank you for your presentation, Eva. It is now time for the Q&A. To ask a question, just type it in where it says type your question here and then press submit at any point right now. So our first question, and this one asks, Eva, how much protein do you use in a sec mouse experiment?
So for the service, we asked the investigator to submit 100 micrograms, and this is very conservative because, as I illustrated, you can measure even in 1 microgram per milliliter concentrations, and usually that represents 9 microliter injection. So this is assuming that the protein is well behaving and eludes well of the sizing column. So I would say conservatively 50 microgram would be my minimum that I would ask investigator to submit to have a good shot at secmoles analysis. But I'm using not the newest generation light scattering detector, so the newest light scattering detector that use the microcell versus my regular flow cell, I think they can do 10 times less protein that I can measure. But I cannot speak for this. I would refer it to Wyatt technology support to ask this question directly for them to get the estimate for the newest detectors.
Excellent, thanks Eva. And our next question asks, do you need to check protein quality before running it on sec mouse? And are there any solutions you wouldn't run, or do you inject any kind of proteins?
So I normally ask people to run size exclusion chromatography in their own labs as the last purification step and submit for secmole analysis fractions directly off the SEC column. I prefer that the protein is not concentrated after SEC, so because those fractions usually are of the best quality. Occasionally, the investigators do not have size exclusion chromatography in their own laboratory. So they submit sample after single-step purification, only the nickel column for the histac proteins, and that's okay, but that is not usually a good quality protein preparation. But if there is nothing else but this, I'll go with it. And occasionally they can be resolved very nicely on the sizing column, and we can characterize individual fractions, but this is less certain. So I prefer at least 2 purification steps. If not SEC, then ion exchange as a secondary purification after affinity column.
Excellent. And are there any solutions you wouldn't run or inject into?
I'm fairly adventurous, I would say. So, I'll usually accept almost any samples that are soluble in aqueous solution at room temperature. But there's a long email exchange before I accept something unusual. So I will research, whether it will not, behave horribly on the sizing column. I'm mainly worried that things will not elute off the column, so there will be nothing to characterize at the end. That's my main worry. Other than that, I usually, I cannot remember even what I've analyzed. I've been doing it for 18 years, so I probably have seen almost anything that you can see.
So it's a lot of samples then?
Yeah, meaning it's a lot and very diverse in terms of, what people send to the service.
And our next question, Eva asks, what is the minimum mass difference that you can reliably detect?
This usually depends on how large is the protein, because the measurements is usually done, we estimate that it has accuracy of 3 to 5% for simple polypeptide, unmodified polypeptides, yes? So if your mass difference is 5%, it's very hard to reliably determine whether, is this mass or the other mass, because the difference is within the accuracy of the measurement, uncertainty. However, if there is a complex and the 2 complexes have very different extinction coefficient, then that allows us to measure something with smaller mass differential, because we can differentiate the 2 possible molecular assemblies by their extinction coefficient. So, if we can have molar mass of 100 and 105, but the extinction coefficient for that 100 molecular weight assembly is 2 times bigger than 105 kilodalton assembly, then we can capitalize on the difference in the UV to RI ratio to differentiate, even if the mass difference is not that great. But typically, I would say at least 15 to 20% mass difference. That's fairly certain that we can measure one versus the other.
Great, thanks, Eva. And our next question asks, does the concentration of the samples that you've mentioned in your presentation, does it refer to the concentration of the injected sample, or is it the local concentration at the peak apex?
It's the eluting concentration at the apex. And to give people some idea, I have a 900 microliter loop on the system. So if I inject 900 microliter of the sample, the eluting concentration is usually at the apex, almost the same as the injecting concentration. So if you inject 100 microliter, then it's usually 10-fold dilution, and so forth. 50 microliter is a 20-fold dilution, so that gives you the estimates of what will be eluting off the column, assuming decent 90% recovery.
Great, thanks, Eva. And just a quick reminder to our live audience, you still have time to ask a question. To ask one, all you need to do is type it in where it says type your question here and then press submit. Our next question, Eva, asks, what type of columns do you usually use? So my preferred column are dextran-based superdexes. Either superdex, I have all of them, peptide, S75, S200, and Super 6
column, which covers a wide range of molecular weight sizing. I also use silica-based column if that's preferred, and sometimes if people have very unusual project and they have developed SEC method in their own lab, I ask them to ship their preferred column to me, and I analyze their sample on their column, because this way I know that the column already have all non-specific sites saturated with the protein of interest. So my SEC trace should look exactly like whatever they see in the lab. So that gives me good confidence that I will have material eluting off the column for analysis. So I use everything, because for sugars, like if I analyze polysugars, I don't remember what are the sugar columns, but there are other columns that I use. So it's driven by the project, really. But for protein, I prefer SuperDexes. Those are my columns of choice.
Excellent. Thanks, Eva. And our next question asks, how do you determine the fractional ratio in DLS experiments?
So this is combination of both DLS measurement to measure hydrodynamic radius and static light scattering measurement to measure molecular weight, because one needs both of these to compute the frictional ratio. Because from molar mass, one can compute what would be the radius of the sphere with the same mass and density, and then you compare this radius to the hydrodynamic radius that was measured from the DLS. So it's needed both measurements to determine the molecular weight and to determine hydrodynamic size.
Great, thanks, Eva. And our next question asks, can you measure small peptides in SEC mouse or large protein assemblies such as virus-like particles?
Absolutely, and that's the beauty of it. Meaning, I measured peptides that I didn't show it, unfortunately, maybe I should have, down to 1 kilodalton. I think even 800 daltons was the smallest one. The trick is that for such small molecules, the concentration has to be relatively high, because the signal in static light scattering detector is proportional to the product of molecular weight and concentration. So if the molecular weight goes down, the concentration has to go up for a limit of detection, obviously. And for virus-like particle, absolutely, we characterize those as well here in the resource. The limit is that the size of the particle cannot be higher than the wavelength of the laser that it used in the light scattering detector. So currently that's around 500 microns, 500 nanometer, which is half a micron in size. In molar mass, probably megadaltons, like hundreds megadaltons.
Excellent, thanks, Eva. And our next question asks, what, in your opinion, is the primary benefit of SEC mouse?
That my understanding is that you measure molecular weight independently of the elution position, and that it can analyze modified proteins with the ease of using the tree detection method. And the concentration range that you can access just on a single system, ranging from 1 microgram to 10 milligrams per milliliter, I think that's relatively remarkable, that the dynamic range of the system is so big, and that you can characterize anything from a peptide of few hundred kilodaltons to the particle that has few hundred megadaltons in size. So, and meaning I hope that the case is illustrated that we can really characterize almost anything on that system in terms of biological samples.
Excellent, thanks, Eva. And I think we've got time for just one last question. And this one starts off as saying, the KD of dimers can only be determined for fast reversible equilibrium complexes, and that's not the case for all complexes. Is there a way to determine KD for other complexes by SEC mouse?
Meaning I have never done it, and
moles would not be my method of choice. If the system is more complex and the assembly is beyond the dimerization, the method of choice would be the composition gradient coupled to light scattering detection, because that's more versatile system, and that would be the technology of choice if I were to do it, rather than moles.
Great. Thanks, Eva. Well, I'm afraid that is all we have time for today. Before you leave us, we would really appreciate if you can spare a few seconds to fill in a very quick survey on your experience today. I would now like to thank Dr. Eva Fulter-Stognew for her presentation and for answering your questions. I would also like to thank the webcast sponsors, Biotechnology, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again for our next webcast.